Okay, so with the previous midpoint presentation, we investigated fun, fresh, and funky trends. This time we're looking at fun, funky, and fresh trends in genetic engineering. Basically, this entire project is mainly focusing on genetic attribution by breaking down a certain database called AdGene. Genetic attribution is looking at a certain sequence to determine where it came from, and there's multiple ways of doing this. There is sort of qualitative ways of doing it, where you might have intelligence agencies or potentially knowledge about events that are happening elsewhere to determine what's most risky. And then there's genetic forensics, which is called microbial forensics in the UK's biosecurity report for this year. But that's a component of genetic attribution where you look at a sequence, and from that sequence alone and no other information can you determine what lab it came from. It's something that I think is growing in popularity as well. The UK's biosecurity report talked about this a good amount and said that the UK needs to focus on improving its capabilities. That's pretty cool. It's generally done with deep learning models. There's the problem here about we don't know how long these are going to be viable, in large part because we don't know how they work, and also because no testing has been done on temporally restricting data and then having them predict future data. So that's what this project aims to do. We also have a positive side effect of being able to analyze a database called AdGene, which has most genetically engineered sequences submitted to it just for safekeeping and collaboration between scientists. And so we can identify regions or countries of concern to direct our attention toward. Theory of change here is that genetic attribution by Mr. Lewis, who is here, or Dr. Lewis. Anyway, the theory of change is that genetic attribution has three different impacts on biosecurity. The first is it lets you identify who created a engineered pathogen and reveal why they might have done it or how to prevent it. There's also accountability, where which I think goes hand in hand with deterrence, where states or individuals or groups that don't want to be held accountable for this might just not create it in the first place, or a state that does it and it doesn't necessarily cause an existential risk, but it does cause a lot of harm. The state can still be held liable to the damages caused. It could also be useful in determining if there's like a lab leak, which lab needs to be held accountable and then which lab needs improvement. And then we also have the analysis of the database, which can help with identification of regions that are most likely to have some sort of biosecurity threat, and also preparation because we now know the regions. And the ultimate goal is to inform subsequent research projects, policy, or industry recommendations. All right, so section one is we're breaking down the AdGene database. The first way that we do this is we can look at sequence length over time, and then we view three somewhat distinct but not that distinct regions. And these are just identified qualitatively. But the first is you have a period of adoption going to about 1995, where there's a lot of variation between sequences and nobody really knows what they're doing, is what it seems like from this data. And we're using sequence length as a sort of proxy for sequence complexity. Then there seems to be some maturation stage where plasmids are getting slightly longer over time, but everything seems to be staying about consistent. And then after around the advent of CRISPR, we don't know if it's because of CRISPR, but it happened around that time. There's a sort of proliferation stage that we called it, where the sequence length grows a lot, and that could signal that there's a lot of new technologies being used, and the sequences themselves are becoming more complex as well. We then broke it down a little bit by lab and country. So breaking it down by lab shows us that it's a pretty diverse data set, with the vast majority of labs submitting a proportionally few amount of plasmids. There's some outliers, but even the biggest outlier is still less than 3%. And then you can also break it down by country, which I think has more interesting implications here. You can view that of the top 5 submitters to AdGene are also 6 or 5 of the 6 largest economies. You have the US, which is number 1, UK number 6, China number 2, Germany number 4, and Japan number 3 or something. But yeah, no, so we can put that on a nice little scatter plot. There you go. This is very preliminary data. You'll also notice that we only have 52,000 plasmids here, but we have 156,000 in the left one, and that's just because I kept getting rate limited by the API, so I can only query like a certain amount per week. That's a problem for the future. So we have we have two fun graphs here. Both kind of look like the data is made up just because it's so consistent. But every single year, there seems to be more unique labs that are depositing to AdGene and also more plasmids that are deposited to the database as well. And because this data seems so clean, we can fit a nice little line through it. And the purpose of this is kind of just like inform some policy recommendations if someone is looking at this and maybe they're not as inclined to view it from a sort of scientific perspective. But basically, we're assuming that AdGene is going to reach 1 million by the year 2045. And the rate is exponential as well. This is a pretty perfect fit through the points. I think it was more obvious when you don't have this extra section, but it goes through the points quite well. Now, to actually analyze the database, we use two different methods. The first are using short k-mers, which is basically a sliding window down DNA sequence. So, for example, here we're looking at 3-mers. You would have ATG, and then you'd have TGG. And then we can also look at codons, which are always 3-mers, but not all 3-mers are codons, just because there's no overlap, and codons only occur in coding regions of DNA. But these code for the actual amino acids that are going to determine the function of whatever protein is being created. It's important to note that these also, that the codons and the k-mers, the trends through all of them appear to be about the same. We see the same trends that we do in 3-mers as 4-mers, as 5-mers, and codons as well. But I'm just going to kind of focus on different ones, and each one, the implication is the same for the other k-mers or codons. In this case, when we look at standard deviation, we see that the database seems to be becoming more homogeneous over time, with the differences between the codons becoming less significant, or the standard deviation is going down, in other words. You can also view some sustained differences in the 3-mers and the 4-mers and the codon proportions as well over time, where, for example, this codon on the left is on the top basically the entire time, and this codon stays at, or this 3-mer stays at the bottom. Same thing with the 4-mers. You can also look at codons, and with codons, we also have a special ability to look at organisms, and organisms have their own codon preferences. And so you can subtract the organism from the codon proportions that you see in the plasmid and determine if it is likely to be naturally originating or from that organism. We did that with a subset of mostly Salmonella, which isn't really applicable. The goal is to do this for every single different model species that's being used, and create this to see if we can actually use it to determine if a sequence is genetically engineered at all. It is pretty interesting, though, that we do see, when we just compare this with Salmonella, that the differences in which ones are overrepresented in genetically engineered ones and which ones are underrepresented, they both code for the amino acid, so it shows some sort of different preference between genetic engineers and Salmonella bacteria. I think also the codon proportion approach is pretty novel. It hasn't really been done in the past. The most successful models have focused on looking at long k-mers that are 18 or longer. And these are pretty useful to identify a lab, just because if a lab has it, then usually no other lab has it. So it can be identified as a sort of signature k-mer. There's also sequence alignment methods, and there's deep learning methods. But we just looked at codon proportions between labs. This will compare the plasmids that each lab created to the same plasmids from another lab, and get the average distance, or the average difference, in other words, between those two labs, and kind of plot it. It is interesting then that the brighter lines mean that the labs are more different from every other lab, and the darker ones mean that the labs are more similar to other labs. It could also be used to identify similarity, and therefore maybe collaborations between labs. Either collaborations that are already acknowledged, or maybe collaborations that labs can be hiding, or maybe that one lab is kind of just like copying off another lab. We can also do a case study on this with two separate labs. Both PIs died, which is why they ended, which is actually interesting. That was not the goal when we chose it. These were just the ones that were depositing for the longest. But you do see the sustained differences between these two labs as well. You can also then use this to train a lot of models. I think the model training is where it gets really interesting. The goal with the model training is also to hand it off to someone else in my mentor's lab, who's going to do more work on the machine learning side of things and see what implications these have. But we do see, for example, that a lot of information is actually held in a sequence length, and using sequence length alone gets you to predicting about 22% of the labs that are depositing plasmids in AdGene. But it really becomes impactful when you add it to something else. So looking at codon proportions, which is the orange line, and that's the second from the bottom, and looking at the codon frequencies, which is the green line that's close to the top. The only difference is that the codon frequencies are the codon proportions multiplied by sequence length. And so when we look at the difference that sequence length there makes, it's pretty huge in determining which lab it came from. You can also use a deep featurizer, which is a more complex method that is oftentimes used more, I think. In this case, we use the DNA deep featurizer, and that did, that's the blue line here. When you combine that with codon proportions, it does even better. But the main implication for this is we can use codon proportions as a proxy for identifying the changes in the data set and implications for genetic engineering attribution. The top models in here, which rely heavily on codon proportions, are actually, it looks like they're probably able to beat the leading interpretable model. And so I think we can use codon proportions as a sort of interpretable measure for complexity. Next steps, we want to get a perspective article from this around April 2024. And in order to do that, we have to tell a story. So we're going to keep breaking down AdGene and analyzing labs. And we are going to look at if we can determine collaboration between labs, which countries are growing their bioeconomies the most. So we had this donut plot before of the proportions of plasmids that were created by different countries, but looking at how that's changing is probably going to be more important. And then we can restrict the data temporally to see if models can be used for future predictions or what proxies we can use for genetic engineering attribution. With that, yeah, thank you to my mentor, the ERA team, Oscar, Delaney, Hannah, Gideon. Yeah, and I'll take questions.